real-time PCR or any PCR, what we do is that we see the twofold increase after every cycle. So how you can understand is that after every cycle, your copy number will be doubled, right? If you see in the graph in the above part of the slide, you will see that as the cycle number is increasing, your copy number is increasing, and suddenly after 20 to 25 cycle number, your number of your copy number keeps on increasing in the exponential manner. If the aim is to check the endpoint detection, mostly in the case of conventional PCR, that is when you have to run the gel in the last moment, you are not bothered to check in between what is happening. So you are not bothered about the real-time detection of the amplification of the gene. So in that case, this particular cycle is okay for you, because at the end you will be able to see the expression or the copy number into the gel. However, if you are concerned about the real-time amplification of the gene, what you need to do is that you have to convert your y-axis into the logarithmic scale. Now, once you convert your y-axis into the logarithmic scale, you will see a linear progression of the amplification in the PCR. So that has been showed in the lower part of the graph in the presentation, right? In this particular presentation. I will not go in detail that what PCR is, and I will just directly go that what are the differences between the real time and the conventional PCR. So basically you have two differences. One is you use for gene expression, you use RNA as a template. So basically you extract the RNA, mRNA, you convert it into the cDNA, and then you amplify it with the PCR. So instead of using DNA, you are using RNA as a template for the gene expression studies. The second variant, and then you will see that you have to use your primer, DNA polymerase, DNTP, buffer, cofactors, and whatsoever is required to actually do the PCR. The second thing what you need is a fluorescent molecule in order to track the real-time amplification. Two of the most common fluorescent molecule is cybergreen and the Tachman probe, right? We will in the today's presentation we will see how the cybergreen works, and Tachman probes will be discussed on the day 3 by Dr. Vinu, where it is mostly used for the pathogen detection. Now, the cybergreen is a molecule which is having aromatic aromatic ring with an elongated chain of propyl and the dimethyl aminopropyl. What it does is that it intercalates in the double-stranded DNA, and it goes and binds in the minor groove of the double-stranded DNA. Once it goes and intercalates between the double-stranded DNA, the fluorescence enhanced by close to 1000-fold. So it means that initially you have some of the fluorescence of the cybergreen. However, once it is adding to the DNA, the fluorescence enhanced to almost 1000 times. So that tells you that how much is the amplification going on, or how much is the number of amplification of copy number is happening. In this particular picture, what you can see is that after the denaturation step, when there is annealing of primer, and then at the time of extension, what happens is that once there is extension, these fluorescent molecules will go and intercalates between the double-stranded DNA. So what happens is that as much as the double-stranded molecules or the double-stranded DNA will be formed, this cybergreen will go and bind into those many number of double-stranded DNA. Eventually, the increase in the fluorescence will be detected. So that can be detected by the light source in the real-time PCR, where there is an excitation of the light, and then after the excitation there is a certain emission at a particular wavelength, which can be monitored by the CCD camera. Okay? So that can be detected by the CCD camera. Now, after that, what you can see is, you can see in your monitor, in your computer, you will see the amplification curve. We will see what this amplification curve is. As I already told, that as soon as the number of the gene amplify, or the copy number is increasing, it will be reflected into the graph, into the normal regular graph. In this graph, you can see that up to around 25 to 26 cycle, you have some certain copy number, but then eventually from 27, 28 cycle, you will see the exponential graph, right? However, it is difficult in the real-time amplification curve to detect the presence in the regular curve. So, as I said, that what we do is that we convert it into the logarithmic scale. Once you convert the graph into the logarithmic scale, now it is important to note that both the graph in the upper part of the slide or in the lower part of the slide is exactly same. Only difference is the y-axis. So in the y-axis you have the direct the fluorescence intensity, and in the below part you have a logarithmic converted y-axis. So what you will notice is that the threshold line, which I will just explain in the next slide, it is too below into the graph, which is difficult to analyze. However, once you convert it into the logarithmic scale, there is a linear phase, and generally for the gene expression purpose, we look into the linear phase of the amplification curve in this particular place. So what this red line is and what this green line is, we will see and understand here in a better way, is that there is a threshold line. It means the minimum fluorescence value which starts detecting, neglecting the background fluorescence, and that line we called as threshold line. The cycle number or the copy number which gives the intensity, fluorescence intensity, which can be measured in some certain number of, after certain number of PCR cycle. So this PCR cycle, where the threshold fluorescence intensity is crossing the threshold line, that is called as the CT. That is the threshold cycle number or CT value. So this CT value is the cycle number where the fluorescence crossed the threshold line. And then in this particular picture, you can see that there is an exponential phase. It means the fluorescence is increasing exponentially. However, after reaching a certain fluorescence intensity, you will see a plateau phase where the fluorescence is not increasing in the exponential manner, and that could be mostly because your PCR ingredients are over in the PCR reaction, and also the shelf lifetime of the cyber grade. So all those things are completely consumed, and then it is not exponentially growing. So you are reaching a plateau phase. So it is very important in the real-time PCR to understand that where you have to select the threshold line. This you can by yourself define where you want to keep your threshold line, or you can leave it automatically based on the PCR machine you are working with, right? So, but it is important that it has to be in a linear path or a linear line of the exponential curve. So, in this particular place, why it is important that where you want to keep your threshold? Let us suppose that this yellow line you plot it somewhere here. What will happen? If you see that this particular PCR, reaction, it's crossing this particular amplification at this point. So almost around 33 cycle. So if you keep your threshold limit at around here, it will not tell you the exact number of copy number present in the initial sample, because the fluorescence is for which if the ingredient is over in the first reaction, it will come into the plateau phase uh, in a much earlier stage than the other reaction. So it is important to keep the threshold line in the linear phase of the exponential graph. So the same thing, this graph is represented here in the second one. in the logarithmic scale. Now you can see in a much better way that you have the amplification curve in a linear graph in this particular image. So that is the importance of having a logarithmic scale in the y-axis, and to keep the threshold line in a linear phase, right? So I hope that it is clear that the aspect what we need to understand, the whole concept for the real-time PCR to understand the gene expression is to evaluate the cycle number where the threshold fluorescence value is exceeding, right? So the fluorescence intensity is exceeding the threshold line. So that particular cycle number we count. Now, in this particular example, you will see that there are three reactions A, B, and C. You will see that the reaction A is reaching the threshold limit at around 23 cycle, whereas reaction B is reaching around 27 cycle, whereas the reaction C is reaching somewhere around the 29 cycle. So what it represents? It represents that if initially if you have the low copy number, it will take more time to reach the threshold value. So if the expression is high, you will reach the threshold limit much before than in the reaction or in the condition where the expression is low. So basically, if the expression is low, you will have the high CT number, CT value. If the expression is high, you will have the low CT value, right? So it goes in this way. In the green box, you will see that the low initial copy number is equals to high CT value, and that is low expression. If the high initial copy number, that is the low CT value, and that is the high expression values, right? One more thing is to understand is mostly tenfold dilution, right, corresponds to a difference of 3.3 cycle. So if you are taking a solution A, and solution A, it means that sample A, and then if you are diluting it to 10 times, so let us suppose that initially you have 100 copy number and you diluted it 10 times, it means now you have 10 copy number. The difference what you would be observing in the real-time PCR is 3.3 cycles apart, and that helps us to calculate the standard curve which we'll be seeing soon, right?